My preceptor read it. Didn't want me to leave. I was like, this.
Okay, so this is a think a nice diagram kind of summarizing that carbonic anhydrase and its role in extracellular buffering. There's just a couple of things that I want to say here. So with respect to the intracellular pH buffering, now as the bicarbonate dissociates, or excuse me, as the carbonic acid dissociates into bicarbonates, it releases a proton, and this proton, if it's just there in space, it's going to acidify the cytoplasm of the red blood cell. But it can be buffered by joining to hemoglobin to make this protonated hemoglobin. Now, this protonated hemoglobin has less affinity for oxygen than the unprotonated hemoglobin. Okay, so kind of put that in the back of your head. Then another thing to put in the back of your head is that much of this carbon dioxide that enters into a red blood cell, or not much, but about a fourth of the carbon dioxide that enters into the red blood cell is not substrate for the carbonic anhydrase reaction, but it's actually carbamating the hemoglobin protein. Okay, so it's making excess carbamation onto the N-terminal end of the 4 hemoglobin monomers. Okay, so let's see, extracellular buffering is by bicarbonate. Intracellular pH buffering is through protein and phosphate. Okay, so let's look at this in the context of breathing, right? Because red blood cells don't just stay in the tissue and pick up carbon dioxide. They go from the tissues to the lungs and back and forth. Okay, so in the tissues, this is where the carbon dioxide, Gesundheit, is going to be produced, right? So the carbon dioxide, a respiring cell is going to be generating carbon dioxide. That enters the red blood cell, produces carbonic acid through the carbonic anhydrase, and then bicarbonate. The proton then, from the dissociation of the carbonic acid, this is going to be protonating hemoglobin and decreasing its affinity for oxygen. So this is referred to as the Bohr effect. But as the pH decreases, the hemoglobin's affinity for oxygen also decreases. And in the case of being, deep into the tissue, this is going to allow oxygenated hemoglobin to deliver its oxygen out to the tissue. Okay. If we're in the lung environment is high in oxygen concentration, and that's going to drive the reaction backwards. Okay, so there's one more point I'll elaborate on here in just a moment. So in the lungs, we have high oxygen concentration, and that's going to push that protonation of hemoglobin in reverse. Okay, so in the high oxygen concentration of the lungs, the oxygen is going to be displacing the proton on hemoglobin. That proton is then going to be a substrate for that reversible carbonic anhydrase reaction to generate the that proton will then, through equilibrium, generate more carbonic acid from bicarbonate. Then the carbonic anhydrase reaction, which is reversible, will generate carbon dioxide, which is exhaled. Okay, so this is the basics of how we can get oxygen from our lungs into the tissues and then get carbon dioxide from the tissues into the lungs to be exhaled. Now, this bicarbonate that's produced here as a result of the carbonic acid dissociating, this doesn't just stay within the red blood cell during this process. It's actually in the tissues. It's effluxing out of the red blood cell in antiport with a chloride ion. So there's a bicarbonate chloride antiporter that in the low oxygen concentration environment of the tissues, it's going to be importing chloride and taking bicarbonate out. And then in the lungs, the reverse reaction is going to be taking chloride out of the cell and then bicarbonate in. Okay. Okay, so I'm going to segue now into a discussion of hemoglobin. And the idea here, that Bohr effect that we talked about, is one of the things that will alter hemoglobin's affinity for oxygen. Okay, so the concentration of protons or the pH of the environment is one of the things that regulates how tightly oxygen is bound to hemoglobin. Okay, so things that affect the affinity of oxygen for hemoglobin through the Bohr effect, proton concentration, the 2,3-bisphosphoglycerate, which I believe we talked about in our glycolysis lecture back in Foundations, but we'll revisit that. And then the carbamation of hemoglobin through that binding of carbon dioxide to the N-terminal ends of the protein change. Okay, so these are the three things that can affect affinity for oxygen, and I'll talk about these one by one. Okay, the first is the Bohr effect, and so this graph is pretty important that you know how to interpret this. This is a graph showing the fractional saturation of hemoglobin with oxygen at different concentrations of oxygen. Okay, so if you have a very high concentration of oxygen, then 100% of your hemoglobin is going to be bound with oxygen. And if you have a very low concentration of oxygen, then 0% of your hemoglobin is going to be bound to oxygen, right? And then at intermediates, you're going to get some percent of your hemoglobin is going to be bound to oxygen or not, okay? Now, we would think, well, hemoglobin's job is to bind oxygen, right? So we want it to be tightly, have very high affinity for oxygen. That's a desirable thing, right? You would think so, but it's really not, because that binding of oxygen is only part of the story with hemoglobin, right? It needs to bind oxygen in the lungs where oxygen concentrations are high, but when it goes into the tissues, it needs to be able to give up the oxygen to a cell that needs that oxygen for respiration, right? So we need to have two different affinities for oxygen, if you will. One that is effective in the high concentration environment of the lungs, and the other in the tissues. So this line here, this is reflecting the different fractional oxygen saturation curves for hemoglobin at different pHs. Okay? And you'll see that as you get more acid, as the pH decreases, the slope gets shallower or more sigmoidal. So you get a decreased affinity for oxygen, and that allows the hemoglobin to give up that oxygen into the tissues. Okay, is that understandable, or am I explaining that well? Okay. Okay, so this is referred to as the Bohr effect, and it's happening a zillion times every time we breathe. This changing the affinity of hemoglobin for oxygen. Okay, so the second thing that will change the affinity of hemoglobin to oxygen is carbamation, and carbamation. This is a significant but lesser metabolic fate of carbon dioxide as it enters into the red blood cell that can make a covalent bond to the amino terminus of the hemoglobin protein chains and make a carboxylic acid, make kind of a pseudo carboxylic acid end on both sides of the protein. And this then also has the effect of decreasing the affinity of hemoglobin for oxygen, both directly through the carbamation and then indirectly because this carbamation releases a proton and decreases the intracellular pH. So you get a direct effect through the carbamation and then an indirect effect through the Bohr effect. All right, so the final thing that we'll talk about that decreases the affinity of hemoglobin for oxygen is 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate is generated through the Rappaport-Luberin shunt of off of glycolysis, and it's a small molecule that functions to sense when oxygen concentrations are low, and then 2,3-bisphosphoglycerate concentrations increase, and then they have the 2,3-bisphosphoglycerate has the same effect as decreased pH or carbamation in that it makes the hemoglobin more readily release the oxygen in the low oxygen concentration environments of the tissues. So this is kind of a busy slide or hard to understand, but if you look at pure hemoglobin, like, you know, in a in vitro reaction without any 2,3-bisphosphoglycerate, that's shown in the black line, and you get a, you know, slightly sigmoidal curve down at the bottom end here, but it's almost a hyperbolic curve. And then if you add 2,3-bisphosphoglycerate into the system, that shifts the curve to the right and makes it more shallow and makes the hemoglobin able to release the oxygen into the tissues where you would want it. And then if you increase 2,3-bisphosphoglycerate, you could shift this curve further on to the right. Okay, so 2,3-bisphosphoglycerate, that's a metabolic response to hypoxia. So, you know, we get that in transient hypoxia, conditions increase. In people who live at high altitudes or people who smoke cigarettes, the 2,3-bisphosphoglycerate levels will be higher as a compensation for that kind of chronic hypoxia. Okay, questions about hemoglobin affinity for oxygen? Okay. Yeah.
So you said the high altitudes, is this also like normal physiological if somebody's like exercising or something? Assuming O2?
Yes, but it's going to be a transient response rather than a chronic response. You know, so yeah, it's whether it would be measurable or not is going to depend on how much that exercise is going to be sustained. Good question. Okay, so let's look at that Rappaport-Luberin shunt and kind of in the context of the whole metabolism of the red blood cell. So what does the red blood cell do for a living? Well, the mature red blood cell doesn't have any mitochondria, so that kind of, you know, eliminates a lot of metabolic possibilities, right? So really the way that the red blood cell is making its energy budgets for the day is through anaerobic glycolysis. So conversion of glucose first to pyruvate and then to lactate through lactate dehydrogenase. This lactate dehydrogenase, this reaction is critical for oxidizing NADH to make NAD+, and this NAD+ that's generated at this step, this is the substrate for the glyceraldehyde 3-phosphate dehydrogenase reaction. Okay, so if we didn't have the lactate dehydrogenase reaction down here, we would basically choke off glycolysis before we had any ATP generation. Okay. Now, so the basics of glycolysis are, you know, north to south there, but we have a couple of east to west diversions that are important. So the Rappaport-Luberin shunt is one that is exists just solely to produce that 2,3-bisphosphoglycerate, which acts as a regulator of hemoglobin. We also so that's kind of one arm off of glycolysis. Then the other two arms off of glycolysis that are significant in a red blood cell have to do with antioxidant defenses. So there's the cytochrome B5 hemoglobin reductase, which is another way that NADH can get oxidized besides the lactate dehydrogenase reaction. And then there is the pentose phosphate pathway, which generates NADPH, which sustains our pool of reduced glutathione within the cells. Okay, so let me remind myself where we are here. Okay, so this is like, it, you know, if you can remember this about glycolysis, I think that gets you a long ways. Basically, in anaerobic conditions, NAD plus needs to be regenerated through lactate dehydrogenase. Okay, simple enough. In terms of that regulation of glycolysis in red blood cells, the regulation is the same as in other cells, where you get inhibition by the ATP and citrates, and then activation by AMP and 2,6 bisphosphol, excuse me, 2,6 fructose 2,6 bisphosphate. Apologize for that. I'm getting mixed up here. Okay, so just reminding you of the key regulatory step, and do with that what you will. Okay, so that Rappaport lubricant shunt is shown here, and that again, it's rather than the 1,3-bisphosphoglycerates going through the ATP generating step of the phosphoglycerate kinase, there is a mutase step that rearranges one of the phosphates to make the 2,3-bisphosphoglycerate. So essentially, the regulation of this pathway is in a cell that is deprived of oxygen. You'll have a buildup of the 3-phosphoglycerates, and that's going to increase 1,3-bisphosphoglycerates and then all of the other upstream intermediates in glycolysis. So that's going to then divert the 1,3-bisphosphoglycerates to more through that mutase pathway to generate the 2,3-bisphosphoglycerate. So the 2,3-bisphosphoglycerate, that does its job with regulating hemoglobin, oxygen affinity. There is a phosphatase that will clip off one of the phosphates to make 3-phosphoglycerate, which can then rejoin glycolysis and ultimately generate an ATP. So if you are into counting high energy phosphate bonds, if something goes through the Rappaport lubricant shunt, you're bypassing an ATP generating step. Okay, so does it matter? Not really. Okay. Okay, so the Rappaport lubricant shunt is about making this regulator of oxygen affinity for hemoglobin. The other two shunts are really about antioxidant defense. So the pentose phosphate pathway, the two major products of the pentose phosphate pathway are 5-carbon sugars, which in most cells would be used for nucleotide biosynthesis. So 5-carbon sugars and then NADPH, right? And NADPH is used for antioxidant defenses to sustain reduced glutathione pools. And it can also be used for biosynthetic reactions. Right? Now, in a mature red blood cell, there's no nucleus, so the requirement for nucleotides is very minimal, right? We need some ATP around, right? But otherwise, you're not going to be, it's not dividing. It doesn't have a high requirement for nucleotide synthesis. So really, the 5-carbon sugar products of the pentose phosphate pathway in a red blood cell, those are going to be going through the regenerative phase, the transketolase and transaldolase reactions to convert the 5-carbon sugars into either 6-carbon or 3-carbon intermediates of glycolysis. Okay, so really we don't do a lot of this in a red blood cell because we don't need to. Okay. Likewise, the NADPH in a red blood cell, the red blood cell doesn't have a real requirement for lipids synthesis or fatty acid biosynthesis, right? It's kind of made and then it's going to just do its thing until it dies. It doesn't really replace its lipids. So the biosynthetic functions of NADPH are not really relevant to a red blood cell. Really what the pentose phosphate pathway is doing in a red blood cell is making NADPH to sustain that pool of reduced glutathione, which is critical for antioxidant defenses within the cell. Okay, so that red blood cell antioxidant defense, is dependent on the pentose phosphate pathway. So there's two NADPH generating steps in the oxidative phase. Just shown here is the glucose 6-phosphate dehydrogenase step. So the glucose 6-phosphate dehydrogenase, this generates NADPH. That NADPH then is a reducing power or provides reducing power for glutathione reductase, which takes, as its name suggests, takes the oxidized form of glutathione and reduces it to the reduced form of glutathione. This reduced form of glutathione then, this provides reducing power for glutathione peroxidase, which is the major neutralizer of hydrogen peroxide, one of the major forms of antioxidants, or excuse me, one of the major oxidant stresses within the cells. Okay, so the elimination of this hydrogen peroxide, this is dependent on some fraction of the carbohydrate that's entering into the red blood cell being diverted through the pentose phosphate pathway. Okay, so this hydrogen peroxide, this can be generated from superoxide or other reactions. Because the red blood cell, its job is to carry oxygen around, so it's under a lot of oxidative stress. Okay, it's a high oxygen environment. Okay, so this oxidative stress, this will happen, this occurs just naturally through metabolism, but there are certain transient conditions that can greatly increase it. So viral infections, some drugs like sulfur-containing antibiotics, for example, some things in our diet, like fava beans, they have a sulfur-containing molecules called vicine and covicine, and their metabolism generates a transient, huge increase in oxidative stress. Okay? Now, in most healthy people, they have enough capacity in their pentose phosphate pathway and enough reduced glutathione that they can take that episodic oxidative stress, and it's essentially invisible to them. They don't have any ill effects from it whatsoever. There is, however, a pretty common inborn error of metabolism called glucose 6-phosphate dehydrogenase deficiency. And this is, as the name suggests, this is a deficiency that causes a lack or loss of activity in the glucose 6-phosphate dehydrogenase enzyme of the pentose phosphate pathway. It doesn't generally kill the cell or kill the enzyme, but it has a greatly reduced activity relative to wild type. Okay, and significance in a red blood cell, if the red blood cell has an enzyme that has reduced activity, it doesn't have any capacity to increase transcription of that enzyme, right, or increase translation of that enzyme. It's kind of stuck with what it has. So in people with glucose 6-phosphate dehydrogenase deficiency, the condition really manifests itself in the red blood cells because other tissues in the body, they can compensate for that lost activity by just increasing expression of the enzyme. Okay, so in people with glucose 6-phosphate dehydrogenase deficiency, they can be quite healthy and have no symptoms until they get exposed to this transient oxidative stress. And in this case, then, they don't have that reserve capacity of reduced glutathione, and they can't regenerate the reduced glutathione fast enough to deal with this oxidative stress. So in that case, then, you get a generation of hydroxyl radical, which can cause peroxidation and damage to membrane lipids in the red blood cell and lead to hemolysis. You can also get the generation of methemoglobin. This is an oxidized form of hemoglobin that can first of all, not do its job of delivering oxygen to tissues, and also it can condense and form an occlusion within cells that is referred to as a Heinz body. So I'll talk about that methemoglobin for a moment. Now, methemoglobin is not only observed in people with glucose 6-phosphate dehydrogenase deficiency. Any kind of a big oxidative stress event can cause the development of methemoglobin, and if you get a lot of methemoglobin around, then the patient will become quite hypoxic because that methemoglobin can't deliver oxygen around. Okay, so the methemoglobin, now so the iron in hemoglobin is heme iron, and we'll talk about heme synthesis on Thursday, but hemoglobin is one place where you have heme iron. Cytochrome P450 enzymes are another one, and they're present in some of the electron transport chain proteins. Now, in some of those other functions of heme iron, you get a change in the oxidation state of the iron with a reaction, right? So you get a change from the ferrous iron to the ferric iron and then back again, right? And that's a normal part of the catalysis for those enzymes. With iron in hemoglobin, it doesn't normally change its oxidation state. It stays in the ferrous form or the +2 form. Okay, and the +2 form, is able to bind oxygen and release it appropriately. When there is oxidative stress within cells, that hemoglobin, the iron can be oxidized to the ferric form or the +3 form, and that's what we refer to as methemoglobin. It's not addition of a methyl group, it's a change of the oxidation state of the iron. So I'm not sure where the nomenclature comes from. But that methemoglobin in this form, it's not capable of delivering oxygen to tissues, and if you get all of your hemoglobin tied up in this form, you've got a real problem of hypoxia. Now the cell, the red blood cell, has that cytochrome B5 methemoglobin reductase enzyme that can take NADH that's generated from glyceraldehyde 3-phosphate and use that as reducing power to convert the ferric iron back into the ferrous iron and essentially restore the hemoglobin to normal. Okay, so if we have sufficient reducing power and if this enzyme is present in the cell, the methemoglobin won't generally become a problem. So with methemoglobin, we can see this in people who have diminished antioxidant responses or defenses, such as a glucose 6-phosphate dehydrogenase deficiency. You can see it in otherwise healthy people when they're exposed to benzocaine is one kind of common anesthetic that will precipitate this methemoglobin crisis in some people. So yeah, it's kind of not exclusive to G6PD deficiency. Okay, and that methemoglobin, that can condense as it's aggregating and accumulating within the cell. It can condense with the oxidized glutathione and other proteins into occlusions or just blobs of non-functional protein, and these are referred to as Heinz bodies, and you can see them. So they're present here and here, and a few of them are not abnormal, but a lot of them are abnormal on a smear. So this is a patient with glucose 6-phosphate dehydrogenase deficiency, and you can see Heinz bodies, and then also bite cells here, and it looks like one here, that are resulting from loss of membrane integrity due to the lipid oxidation. Okay, so people with methemoglobinemia, just if you examine their blood grossly, it's going to look a lot darker than normal blood. And if you have somebody with methemoglobinemia, you can restore the hemoglobin with the ferrous iron, the +2 iron, with a compound called methylene blue. And methylene blue is using NADPH as another way to reduce the ferric iron in methemoglobin to the ferrous iron in normal hemoglobin. So this methylene blue, this is acting as like another electron transfer or another route to get the electrons to the iron. Now, methylene blue is a treatment for methemoglobinemia. However, in people with glucose 6-phosphate dehydrogenase deficiency, it would not be indicated because people with glucose 6-phosphate dehydrogenase deficiency have a diminished capacity to make NADPH, right? So methylene blue is useful for methemoglobinemia, but not in cases of diminished NADPH production. Okay, should we do a few clicker slides? Okay. See how we do here. So you got 2,3-bisphosphoglycerates, protons through the Bohr effect, and then carbamation. Good. Okay, so this would presume that methylene blue would be appropriate for this patient because I'm just looking here, like, would ferric iron, would that be a answer for this as well, maybe? I guess, yeah, okay, technically that would be a good idea. Okay, but methemoglobin's better. All right, so we got a few more questions that kind of touch on or tee up the next topic that we'll discuss. And this is getting real deep into trivialities of foundations, but it's an important drug target, so should know it. Ooh, very good. Ribonucleotide reductase, best answer there. Okay, this is kind of a conceptual question, but just... Okay, JAK-STAT receptor. Very good. All right, last question. Okay, so we're getting a little esoteric with these questions here, right? It's all leading to something. Okay, so what does a Newtonian fluid mean? So a Newtonian fluid means that there's a linear relationship between the amount of, did you answer this one? Or? Okay, it looks like this one isn't working. Okay, so what's a Newtonian fluid? A Newtonian fluid is one where, kind of my simple non-physicist explanation of it, it has a linear relationship between how much the fluid deflects and how much force you put onto it. Okay, now, honey is a Newtonian fluid, right? Okay, and how it moves, I can take a spoon and use my arm muscles and put a big force on it, and it's going to have a big movement away, right? And I can pour it out and put a small force on it, the force of gravity, and it's also going to move, right? But it's going to move slower because it's a smaller mass, right? Okay, now, what about mayonnaise? If I add a big force, we get a big movement, right? But what if I have a small force? If I shook it out, I could get it to blop onto the floor. But a small force, it doesn't have that linear relationship between the force that you put on it and how much it moves. That make sense? Okay, so how nonlinear it is, that is, how nonlinear it is dependent on that difference in the small forces versus the large forces. So blood is acting not like honey, but more like mayonnaise. It's a non-Newtonian fluid. So blood is, as we know now, it's a liquid, but it has these particles suspended in it that are cells, right? Red blood cells and platelets and white blood cells and what have you. Now, if those cells were marbles, say, glass marbles or steel ball bearings, when they collided with one another, there would be a very predictable response based on Newtonian physics that if you hit something at this angle with this force, then they're going to deflect in predictable ways, right? But that's not how cells work. Cells, when they interact with one another, there's all these proteins on the cell surface, and they're all receptors and ligands for one another. So when two cells come together, it's more like a Velcro binding and then Velcro ripping off. There's 1,000 different affinity coefficients, essentially, of these different receptors that are, and the proteins that are studying the surface of the cells, that are making it not a predictable collision, but kind of a little different, anyways, when you have those small forces. So blood is a non-Newtonian fluid. So this is a diagram that's showing, blood versus Newtonian fluids like water. So this is measuring essentially the force that you put on a fluid versus how much it deflects, and that there's a mechanical engineer would probably be able to give you a better explanation from that, but I think it's sufficient. So water and non-cellular portion of blood, right, the plasma, those are behaving in Newtonian fashion, where there's this perfectly linear relationship between how much the liquid moves and the stress that you put on it. So if you take plasma, though, and add back the cells, then you get a linear relationship between force and movement with high forces, but there's this deflection away from linearity at lower forces, and those lower forces, that's more what the body is going to be experiencing in circulation. Okay. So what is this condition called polycythemia? Okay, let's talk about that a little bit. Now, normally blood viscosity is going to be determined and how nonlinear it is, it's going to be determined by how many cells there are, because it's those interactions between the cells that are, making the fluid non-Newtonian. So the more cells there are, the more viscous the fluid is going to be. So what is controlling the amount of cells within the blood? The amount of cells is going to be a balance between the cells produced and the cells lost. Okay, and we'll talk about the cells produced now, and we'll touch on the cells being lost a little bit on Thursday. So the production of red blood cells occurs in the bone marrow through erythroid precursors, and the rate at which those erythroid precursors are proliferating and differentiating into mature red blood cells is sensitive to how much oxygen is within the body. Do you remember HIF-1 alpha and prolyl hydroxylase and all that story? Okay, so I'm not going to go into the details of that, but that system is present in the kidney, and one of the genes that is sensitive to oxygen deprivation through that HIF transcription factor is a protein that's similar to a cytokine called erythropoietin. And erythropoietin is acting as a promoter of red blood cell maturation and differentiation and proliferation at multiple steps in the kind of bone marrow pre-blood cell lineages. And you will talk very shortly about all the different lineages of the blood cells if you haven't by now. So, okay, so erythropoietin, it's acting on these red blood cell precursors in the bone marrow, and it's acting through a receptor complex that is a JAK-STAT receptor. So this is similar to cytokine receptors, so this is kind of a common receptor organization, and I like to think of it as a tyrosine kinase receptor and a steroid receptor having a baby, right? Because there's elements of both. When you get so this would be like a cytokine or erythropoietin or what have you, a ligand, binds to the JAK-STAT receptor. There is a JAK molecule or a JAK protein that stands for Janus kinase that's associated with the intracellular parts of the receptor, and that acts as a tyrosine kinase that's phosphorylating multiple tyrosines on the intracellular portion of the receptor. So that's kind of similar to growth factor receptor or insulin receptor or what have you. Okay, then after that phosphorylation event occurs, then you get something that's similar to estrogen receptor or thyroid hormone receptor or something like that, where you get another portion, a STAT molecule, that dimerizes and then enters into the nucleus and causes increased in transcription of different genes by binding to promoters. Okay, and with respect to the bone marrow precursor cells that are being stimulated by erythropoietin, there are anti-apoptotic genes and pro-proliferation genes like cyclin D1 that are under control of the JAK2 stimulus. Okay, so yeah, this is just kind of a general scheme of how JAK-STAT receptors work, and then the specifics of the erythropoietin receptor. That's a receptor that's complex to two of these JAK2 subunits. Okay. All right, so there are conditions where we have too much red blood cells, and our red blood is too viscous, and that can cause problems with the vasculature, as you can imagine. So polycythemia vera is a condition of elevated red blood cell production due to gain of function mutations in the JAK2 gene. This is showing kind of different combinations of the JAK receptors that can be ligands for different cytokines and what have you. The erythropoietin receptor is this dimer that's linked to JAK2, and there are gain-of-function mutations that cause a essentially constitutively active JAK2 in erythroid precursor cells, and that results in elevated red blood cell production and increased blood viscosity. So there are some inherited mutations that result in the polycythemia vera, and these are autosomal dominance inheritance patterns. And those are much rarer than the majority of the cases, which are autosomal mutation in just some subset of the red blood cell precursors will precipitate this crisis. Okay, that's shown here. How would we treat polycythemia vera?
Bone marrow transplant?
Ooh, if you wanted to cure it,
Okay.
that might be that's a good option. But just to treat it, there are two treatment options, and one is the old standby, phlebotomy. You just let them bleed, and get rid of the excess blood, and you'd have to do that periodically as the hematocrit increased. There's another drug, hydroxyurea, and this is an inhibitor of ribonucleotide reductase, and that's that enzyme that is making a nucleotide into a deoxynucleotide that could be used for DNA. So hydroxyurea is basically decreasing the rate of proliferation of those bone marrow, those red blood cell precursors in the bone marrow, and trying to slow down the rate of their production. Okay. Yes.
So like with phlebotomy, wouldn't you be taking out like plasma too? So then you wouldn't necessarily be changing like the ratio of hemoglobin to plasma, but you would just be overall decreasing volume?
Well, you get a redistribution of volume though, like you get fluid coming from the tissues into the vasculature that's going to dilute out the red blood cells.
That makes sense.
Yeah, good question though. Okay, let's see, one clicker question here. I do have the case, but I don't think we're gonna. Looks like my clicker's kind of stopped working here, but. Okay, so simplified view of metabolism, right? Super easy for me to recycle this figure for an exam question. So which one is a red blood cell, do you think? Let's see. This is the mitochondria. So the cell that's just taking in glucose and effluxing lactate, that's going to be the red blood cell's metabolism. So which one is the hepatocyte? What's a feature of hepatocytes that sets it apart from just about every other cell in the body? The ability to spit out glucose, right? No other cells do that. Okay. All right, so just kind of view cells, in big picture metabolic processes, and know a little bit about the details that kind of promote those big processes. Okay, so I'm going to skip the case. There was a interesting case, and I'll try to tee that up for Thursday's lecture so we can talk about it then. Okay, let me know if you have questions about this material. It's overactive downstream of erythropoietin. It's the receptor for erythropoietin is whether there's erythropoietin.
How this week
Played some
mine's on Thursday.
I would
How would you
It didn't happen. worst group.
I know. I told Sarah.
Did you? I was gonna tell her. You beat me to it. Would you tell Ashley?
I'm right here.
Oh, sorry. Callie, it's up.
Sally, are you here on Friday? Are you here on Friday? Play volleyball. There's like this. My guess is I think it's kind of.
So you're saying I
I'll see you later. Okay, have fun at text you. This out into its place. And it's like an open field. But Sarah, me, and Penny were gonna go. Don't mind my Tupperware.
Oh, that's all right.
It might be, yeah, like similar to, you guys are playing for a long time, and you're like lying down, maybe that's.
Mine just finished.
I cleaned up the tour. That doesn't happen anymore.
Everybody's clean now. That's all it takes.
Yeah, he would just rename it with the on.
Oh, yeah. Hi, Tron, how are you?
Good, you staying for the meeting at noon?
It's kind of hard to like learn the language of it.
It'd be such a nice future.
What am I?
Oh, and this
One of
one.
my kiddos from summer camp. He's not a kiddo anymore. He's wanting to apply to med school. He's like, learn everything now. Use it for your MCAT. Do it. Learn it now.
Okay, I'll be back down.
That wore the one guy only wore it coming into the hospital and then took it out first.
And it does this again. What the hell?
It does what?
This isn't connected because I changed it. This was like 120. And then I changed it like this.
You have 30 details.
All the hospitals wore them. Pretty much.
Yeah. I think it's because
It was
So,
what's
and
wrong
that
with
is
this?
30.
Yeah. I'm confused.
Yeah, I don't know.
Lower coat and tied together.
I hate it. Ugh. Okay, I hate it. Huh? Is there
Ooh.
pizza?
The Duluth Town Hall.
There's no pizza.
I'm not staying then. What are they talking about though?
I mean, you could probably, it depends on what type of town hall meeting it is. Well, there's different types of town hall meetings. Sometimes they have a set agenda and sometimes they have like an open floor that you can bring whatever concerns you have. Sometimes they have like themes.
What's the theme?
I don't know.
Just hijack.
I mean, you can do that. Like some people are not good at
What's the town hall?
meetings.
That's so expensive.
Well, I don't know.
Town
what-
Hall rescheduled. In parentheses.
So this is the rescheduled one. Let me see.
You should go and tell them to get a flashing stoplight at that,
Forget you did tomorrow.
or a flashing pedestrian wall.
That's the wrong town hall. Is that like a city thing, I'm guessing? What the hell is this town hall? Where's Google Maps? Google.
Thank you, Aaron.
I'm gonna look at the Google Calendar real quick, because I don't know.
Share the.
I won't be on the Google